Sugar is processed differently in the brains of obesity-prone versus obesity-resistant rats. Sugar is processed differently in the brains of obesity-prone versus obesity-resistant rats. You're listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting, presented for World Reading Club in association with UniquilibriuM. This edition's reading focus comes to us from neurosciencenews.com and is titled, Sugar is Processed Differently in the Brains of Obesity-Prone versus Obesity-Resistant Rats. It was published on February 3rd, 2023, under the category of Neuroscience. Summary. Glucose takes longer to get into the nucleus accumbens of obesity-prone rat models. Glucose takes longer to get into the nucleus accumbens of obesity-prone rat models. Researchers also discovered excess levels of glutamate in obesity-prone rats, implying a deficiency in the neurotransmitter recycling process normally maintained by astrocytes. Source: University of Michigan. On a diet? Perhaps you're avoiding sweets or carbs altogether or curbing late-night munchies. These are examples of behavior modifications, and when it comes to food, avoiding those diet triggers can be pretty hard to do. To understand what drives people to overeat, scientists are looking more closely at a brain structure involved in motivation, called the nucleus accumbens. This small region drives reward-seeking behavior, underlying the pursuit of sex, recreational drugs like nicotine and alcohol, and food. These brain motivation centers evolved to help us survive. Finding food and having sex are essential to the survival of an individual and of a species, said Carrie Ferrario, Ph.D., Associate Professor in the Department of Pharmacology at Medical School. What was advantageous when food was hard to find has become a disadvantage and unhealthy in the current food-dense environment. This is compounded by the overabundance of overprocessed, low-nutrition foods that may satisfy our taste but leaves our bodies undernourished. People don't tend to find it difficult to turn down an extra serving of broccoli, but just one more French fry or making room for a bit of chocolate dessert, that's a different story. The real challenge is overcoming these urges and changing our behavior when it comes to food, Ferrario added. Given the immense toll obesity takes on virtually all body systems, Ferrario, Peter Volbrecht, Ph.D. of Western Michigan University, and their colleagues are using rat models to understand potential brain differences between animals who are prone to overeating and obesity and those who are not. Previous research from Ferrario's lab pinpointed differences in the nucleus accumbens in obesity-prone and obesity-resistant rats. Their latest study, published in the Journal of Neurochemistry, tracked what was happening in real time in the brain when these animals were presented with glucose, a type of sugar labeled with a tracer. The tracer allowed the researchers to measure this new sugar in the brain. Sugar is the brain's main fuel source, and once there, the molecule is broken down and used to create new molecules such as glutamine, glutamate, and GABA, each with an important role in influencing the activation of neurons in the brain and nervous system. Glucose that is consumed gets broken down, and then its carbon gets incorporated into neurotransmitters. We see those labeled carbons showing up in those molecules. Glutamate, glutamine, and GABA over time, explained Volbrecht. They found that glucose was taking longer to get into the nucleus accumbens of obesity-prone animals. Furthermore, when measuring the concentration of the glutamate, glutamine, and GABA, they discovered excess levels of glutamate, an excitatory transmitter. This, said the team, implied a deficit in a neurotransmitter recycling process, typically maintained in the nervous system by star-shaped cells called astrocytes. Our first featured image here shows a little bit of an analysis of what's going on in the brain here, and I'll just read the caption, which says, Example microdialysis probe placement verification. A fast green dye can be observed at the site of dialysis within the nucleus accumbens, which is shown as a black arrow and green-blue coloration. The track of the probe from the dorsal aspect of the brain to the nucleus accumbens is indicated by the white arrow. The dotted line indicates the rough borders of the nucleus accumbens. Scale bar equals 1 millimeter. Credit the researchers. You can see this featured image here on neurosciencenews.com in the article. Normally, and here we go, Neuroscience News continues. Normally, astrocytes will pull glutamate out of the space between neurons, called the synapse, convert it into glutamine, and then shuttle it back to cells that produce GABA or glutamate. This sequence is crucial for turning neurons off and on. The findings suggest that we're getting too much glutamate and it's not being taken out of the synapse, said Volbrecht. Ferrario added, the balance between glutamate and GABA, the main inhibitory transmitter, is really important for brain function and will influence activity of the neurons in the nucleus accumbens. This balance, and therefore brain activity, is different in obesity-prone versus obesity-resistant rats. The fact that these rats are either prone to obesity or not is important for disentangling cause and effect, says Volbrecht. It allows us to remove diet as one of the variables. The team hopes to next study the role of inflammation in the development of obesity and how differences in brain function contribute to susceptibility and resistance to obesity. Other authors on the paper include Catherine M. Nesbitt, Victoria M. Addis, Keenan M. Bullnamore, Daniel A. Michelli, Kendall B. Smith, Darlene A. Sandoval, and Robert T. Kennedy. About this neuroscience research news. Author, Press Office. Source, University of Michigan. The image referred to earlier is in the public domain, and the original research is a paper which is under open access and is titled Differential Regulation of Nucleus Accumbens, Glutamate and GABA in Obesity-Prone and Obesity-Resistant Rats by Peter J. Volbrecht and others, Journal of Neurochemistry. You've been listening to Technical News Reading, presented by Hakeem Alibokis Alexander on Spreaker, social podcasting, presented for World Reading Club in association with UniquilibriuM. Neuroscience News continues now with an abstract from the paper titled Differential Regulation of Nucleus Accumbens Glutamate and GABA in Obesity-Prone and Obesity-Resistant Rats. Obesity is one of the leading health concerns in the United States. Studies from human and rodent models suggest that inherent differences in the function of brain motivation centers, including the nucleus accumbens, contribute to overeating and thus obesity. For example, there are basal enhancements in the excitability of nucleus accumbens GABAergic medium spiny neurons, and reductions in basal expressions of AMPA-type glutamate receptors in obesity-prone versus obesity-resistant rats. However, very little is known about the regulation of extracellular glutamate and GABA within the nucleus accumbens of these models. Here, we gave obesity-prone and obesity-resistant rats stable isotope-labeled glucose, 13C6 glucose, and used liquid chromatography mass spectrometry LC-MS analysis of the nucleus accumbens dialysate to examine the real-time incorporation of 13C6 glucose into the glutamate, glutamine, and GABA. This novel approach allowed us to identify differences in glucose utilization for neurotransmitter production between these selectively bred lines. We found that voluntarily ingested or gastrically infused 13C6 glucose rapidly enters the nucleus accumbens and is incorporated into 13C2 glutamine, 13C2 glutamate, and 13C2 GABA in both groups within minutes. However, the magnitude of increases in nucleus accumbens 13C2 glutamate and 13C2 GABA were lower in obesity-prone than in obesity-resistant rats, while basal levels of glutamate were elevated. This suggested that there may be differences in the astrocyte regulation of these analytes. Thus, we next examined nucleus accumbens glutamine synthetase, GAD67, and GLT1 protein expression. Consistent with reduced 13C2 glutamine and 13C2 GABA, nucleus accumbens glutamine synthetase and GLT1 protein expression were reduced in obesity-prone versus obesity-resistant groups. Let's start this again. This suggested that there may be differences in the astrocyte regulation of these analytes. Thus, we next examined nucleus accumbens glutamine synthetase, GAD67, and GLT1 protein expression. Consistent with reduced 13C2 glutamine and 13C2 GABA, nucleus accumbens glutamine synthetase and GLT1 protein expression were reduced in obesity-prone versus obesity-resistant groups. Taken together, these data show that nucleus accumbens glucose utilization differs dramatically between obesity-prone and obesity-resistant rats, favoring glutamate over GABA production in obesity-prone rats, and that reductions in nucleus accumbens astrocytic recycling of glutamate contribute to these differences. These data are discussed in light of established differences in nucleus accumbens function between these models and the role of the nucleus accumbens in feeding behavior. NeuroscienceNews.com asks that you join our newsletter. Sign up to receive our recent neuroscience headlines and summaries sent to your email once a day, totally free. We hate spam and only use your email to contact you about newsletters. You can cancel your subscription anytime. Neuroscience News posts science research news from labs, universities, hospitals, and news departments around the world. Science articles can cover neuroscience, psychology, AI, robotics, neurology, brain cancer, mental health, machine learning, autism, Parkinson's, Alzheimer's, brain research, depression, and other topics related to cognitive sciences. You've been listening to Technical News Reading, presented by Hakeem Alibokas Alexander on Spreaker, a social podcasting, presented for World Reading Club in association with UniquilibriuM. This edition's reading focus has come to us from neurosciencenews.com and is titled, Sugar is Processed Differently in the Brains of Obesity-Prone versus Obesity-Resistant Rats. It was published on February 3rd, 2023 under the category of Neuroscience. You can read this article and others like it for yourself by visiting neurosciencenews.com. That's neurosciencenews.com. neurosciencenews.com. Sugar is processed differently in the brains of obesity-prone versus obesity-resistant rats.